Okay, very good afternoon, my dear friends. Today's our eminent resource person, Dr.A.SHYLAJA Madam Garu, Assistant Professor, Department of Botany, Government Degree College, Autonomous, SIDDIPET. Madam is going to explain about one of the important area in plant anatomy, that is stem anatomy, and the primary structure of the stem, at the same time, the secondary growth. Madam, please start your session. Please share your screen.
Okay, so thank you, sir. Good afternoon to all. Today I am going to discuss about the stem primary structure and after that secondary growth. I will share the screen. Sir, it is visible? Sir.
Yes, madam, it's visible. Madam, madam, if possible, please click on slideshow.
Okay, sir.
Okay, madam. Yes. Now it's clearly visible. Yes,
okay,
madam.
Okay.
Yes.
Okay, so anatomy of dicot stem is studied by TS. So in the stem has following regions, that is epidermis, cortex and stele. See here, I am explaining clearly with diagrams also. See here, there is a epidermis, cortex and stele. See here in the epidermis, this is the outermost layer of the stem. Its cells are closely arranged in a single layer. The epidermis cells are closely arranged in a single layer without any intercellular spaces. See here in this epidermis, that is a single layer without any intercellular space, that cells are all arranged. Cuticle is present over the outer surface of the cell walls. So on the epidermis, that cuticle is present over the outer surface of the cell wall. Chloroplasts are absent in this layer. Stomata and multicellular hairs are present. Okay, so that is the epidermis. After we are coming to the cortex. In the cortex, we are observed that hypodermis, outer cortex and inner cortex and endodermis. These parts are present in the cortex. 4 parts are there in the cortex. After that, stele, in the stele, we have observed 3 parts: pericycle, vascular bundles and medullary rays and pith. Okay. So first, see here, this is the epidermis. In this clear picture, you have to see that epidermis and hypodermis is there. It is the outermost layer composed of parenchymatous cells. It is a single layer of cells closely packed. Okay, no intercellular spaces are there in that cells. Outer tangential wall is thick, that is deposited with fatty substances, that is cutin. Okay. That is cuticle or cutin. That is the outer layer of the epidermis. Cutin occurs as separate layer, that is called cuticle. Okay, cutin is occurred on the epidermis, that is as a layer, that is called cuticle. Okay, epidermis of young stem contains stomata. In the young stem, we are observed that on the epidermis, the stomata are present. Okay, see here. Next. We are coming to the next slide. That is the see here, multicellular hair usually present over the epidermis. See here in the picture, you have to observe multicellular hairs are there on the epidermis. See here, these are the multicellular hairs that we are called trichomes. Okay, that is the trichomes. In herbs, epidermis remains throughout the life cycle. In woody plants, epidermis is replaced after the secondary growth. So, functions of epidermis is a protection layer, and the cuticle prevent the water loss. Stomata in stem facilitate gaseous exchange. The stomata present in the epidermis, that is that function of stomata is gaseous exchange. Trichomes and hairs provide the protection. Okay, trichomes or multicellular hairs are present in the epidermis that will be provide protection. Okay, this is the epidermis. Next, we are coming to the cortex. So, in the cortex, the part of the stem present between the epidermis and stele, it is known as cortex. Okay, in between the epidermis and stele, that part is called cortex. It consists of three portions, that is hypodermis, general cortex and endodermis. In the cortex, in the general cortex, that two parts are there, that is the outer cortex and inner cortex, and after that endodermis. These four parts are present in the cortex. The hypodermis is located just below the epidermis and made up of 2 to 6 layers of collenchyma. See here, below the epidermis, that is the 2 to 6 layers of collenchyma cells are present in the below the epidermis, that is called hypodermis. Okay, chloroplast can be seen in these cells. That is the hypodermis. Okay, see here in the next slide. Okay, see here. Below the epidermis, the 2 to 6 layers of collenchyma cells are present that we are called hypodermis. Chloroplast also can be seen in that cells. The general cortex is positioned below the hypodermis and is formed by several layers of parenchyma. And chloroplasts in the outer cells and colorless plastids in the inner cells are present in these cells. And we are observed the two types of cells, that is chloroplasts in the outer cells and colorless plastids are also we are observed in the inner cells. Okay, that innermost layer of the cortex is called the endodermis. So outer layer is filled with the collenchyma and inner cortex is filled with the parenchyma. Okay, remember this. And after that, usually the chloroplasts are absent in the hypodermis. But in the xerophytes, the hypodermis is sclerenchymatous. Generally, in the xerophytes, we are observed that the hypodermis generally in the xerophytes sclerenchymatous, but we are here in the dicot stems, we are observed that is the collenchymatous. Outer cortex cells are filled with the collenchyma cells. Okay, see here. What is the function of hypodermis? That provide the mechanical support in plants with secondary thickening. Hypodermal cells gives rise to cork cambium, which give rise the bark. At the time of secondary growth, we are observed that thickening hypodermal cells are there that gives rise to cork cambium. Okay, that which gives rise the bark. Okay, when we are observed the bark at the secondary growth time, that will be coming from the hypodermal cells. Okay, so see here in this outer cortex is there. Okay, tissue inner to hypodermis, that we are called outer cortex. The cells all are chlorenchymatous, loosely packed cells with the intercellular spaces. Some intercellular spaces are arranged in that cells. In the xerophytes, outer cortical cells form the palisade-like tissue. Okay, so function is the photosynthesis, because of that chloroplasts are there in that cells. Okay, next we are coming to the inner cortex. So cells all are parenchymatous, loosely packed with intercellular spaces. Function is the storage of carbohydrates. See here, carbohydrates in the inner cortex, that all cells are filled with the parenchymatous cells. That is the storage of carbohydrates. Main function of inner cortex is the storage of carbohydrates. So next cortex. In the hydrophytes, generally the cortex with plenty of air cavities. In the xerophytes, there is the sclerenchymatous cells are present in the cortex, in the hypodermis. But in hydrophytes, The cortex is with plenty of air cavities, aerenchyma. Okay, so cavities are there. That is the aerenchyma that helps in the gaseous exchange and provide buoyancy of to plant. Okay, that is the gaseous exchange. So sclerenchymatous patches occurs in the cortex of eucalyptus and eugenia, ficus. Some plants we are observed that sclerenchymatous patches also present. Secretory cavities occurs in the cortex of eucalyptus. Some secretory cavities we are observed in the eucalyptus plant. The resin canals occurs in the cortex of anacardium. In the anacardium, we are observed the resin canals. Lattice-fellow cells occurs in the cortex of the latex producing plants. Latex producing plants, we are observed that lattice cells also present in the some latex plants. That is the cortex. What are the functions of cortex? Mechanical support by the hypodermis, and during the secondary growth, that hypodermal cells gives rise to cork cambium for the bark formation. That is also very, very important. At the time of secondary growth, that all hypodermal cells gives rise to cork cambium. So that cork cambium forms the bark. Okay. At the time of secondary growth, chlorenchymatous cells in the outer cortex can do photosynthesis. Some cells having the chloroplast, so that's why that doing the photosynthesis. Parenchymatous cells of inner cortex can storage carbohydrates. These all are functions of cortex. Cortical cells also stores the ergastic substances, some latex, resin canals, latex canals. Okay, we are observed in the cortex. These all are functions of cortex. Okay, so next we are coming to the endodermis. So epidermis and cortex and stele. So that endodermis is present outer layer of the cortex. Okay, or stele. So exact function of endodermis is not known. They do not allow the passage of water, can storage food material as starch grains. Okay, so that is the endodermis. Next we are coming to the stele. In the endodermis, the cells are arranged in a single layer. Casparian strips are present in the endodermis. After that stele, it is a central vascular cylinder. See here. In the stele, we are observed that pericycle, vascular bundle, medullary rays, pith. Okay, pericycle is the stele outermost layer. Okay, so see here in the vascular bundles, that outermost layer of the stele is called pericycle, and cortex last layer is called endodermis. Okay, outermost layer of the stele is called pericycle. It located next to endodermis. After the endodermis, that will be present pericycle. Show wide variation. So in some plants, pericycle is absent. Some only we are observed in the some plants only. Usually multi-layered, but 3 or more pericycles may be completely parenchymatous or completely sclerenchymatous. So mixture of parenchyma and sclerenchyma also, alternating bands also present in some plants. We are observed that some different types of cells are there in the pericycle. After we are in the we are discussed in the last class, that is in the root anatomy, the pericycle helps in the lateral roots are arised from the pericycle. Okay, so in the vascular bundle, next we are coming to the vascular bundle, also called as fascicles, located inner to pericycle, developed from the procambium. So see here, arranged as a broken ring. See here. See here. In the vascular bundles are metaxylem and protoxylem. Conjoint, xylem and phloem together, that we are called conjoint. Open with cambium. Okay, open type of vascular bundles means in between the xylem and phloem, that cambium is present. That vascular bundles are called open type of vascular bundles. Xylem endark means protoxylem towards the interior. Okay, that protoxylem is formed as first, that is called a protoxylem, that towards the interior, that is they are called xylem endark, collateral and or bicollateral. Okay, Collateral usual type, one patch of xylem and one patch of phloem. Okay, that is the collateral. Bicollateral means a median patch of xylem laying in between two phloem patches. Between the two phloem patches, the xylem is there, that we are called bicollateral. Okay, so bicollateral or a characteristic of Cucurbitaceae. In the Cucurbitaceae family, we are observed that bicollateral vascular bundles are present. In that vascular bundles, xylem, it helps in the water and minerals conducting tissue of vascular bundle. Okay, water and minerals are conducting tissue of vascular bundle, that is the xylem. It composed of tracheids, vessels, fibers, and parenchyma. Okay, we are observed in the xylem. These are xylem elements: tracheids, vessels, fibers, and parenchyma. Xylem differentiated into protoxylem and metaxylem. What is the protoxylem or what is the metaxylem? See here. First formed part of xylem is called protoxylem. Arranged towards the center, very less amount of tracheary element, large amount of parenchyma. Large amount of parenchyma is present in the xylem, and tracheary elements with a narrow lumen, annular or spiral thickenings are present in the protoxylem. See here. So what is the protoxylem? First formed xylem is called protoxylem. After that mature stage, okay, formed after protoxylem, that is called metaxylem. Okay. So formed after protoxylem, more tracheary elements than protoxylem, cells with large lumen, cells with reticular or pictured thickenings. Okay, thickenings are present in the metaxylem. And next, we are coming to the xylem after phloem. Okay, xylem is a water conducting tissue and phloem is a food conducting tissue of vascular bundle. It composed of sieve tubes, companion cells, parenchyma, and fibers. Okay, sieve tubes, companion cells, parenchyma, and fibers. So it is also differentiated into protophlloem, metaphlloem. Protophlloem means first formed, arranged towards the periphery. Metaphloem, same, differentiated after protophlloem, located near to cambium. That is the metaphloem. Okay. So what is the cambium? See here in this diagram very clearly I'm showing that the cambium in between the xylem and phloem, that is called cambium. A layer of meristematic tissue is present between the xylem and phloem, that is called cambium, meristematic tissue. Okay. This is also called fascicular cambium or vesicular cambium. It is a remnant of original procambium. Cells are parenchymatous and thin-walled. The cells are parenchymatous and thin-walled. Cells with dense cytoplasm and a prominent nucleus. So, in the secondary growth of dicots occurs due to the cambium. Only that it present in between the xylem and phloem. At the time of secondary growth, we are observed various parts are arranged by the cambium. Okay, the vascular bundle with cambium is called open vascular bundle. Okay, so see here next medullary rays. This is also called a pith ray. It is in between the vascular bundle space is called a pith ray or medullary ray. Occurs in between of vascular bundles, composed of parenchymatous cells. So it is also composed with the parenchymatous cells. Cells are radially elongated. During the secondary growth, the cells of the medullary rays it gives rise to interfascicular cambium. Okay, so these are medullary rays all are arranged with at the time of secondary growth. Cells of the medullary rays gives rise to interfascicular cambium. It formed that interfascicular cambium is very important. Medullary rays or the cells of the medullary rays gives rise to interfascicular cambium. Okay, functions allow radial transport of water, provide interfascicular cambium during the secondary growth. It's very important. Medullary ray functions. What is the function of medullary ray? It allowed radial transport of water and it provides the interfascicular cambium at the time of secondary growth. That is a medullary ray function. And after that pith, okay, see here. This is also called a medulla and located towards the inner side of the vascular bundle, exact central portion of the stem. that is called a pith. Okay, mostly parenchymatous. The pith is mostly parenchymatous. Parenchyma may be loosely arranged. Parenchyma may be loosely arranged. Sometimes wall thickening also occurs in that. Cell of outer region smaller and inner region is larger. That is the pith. Okay. So see here, that is in this diagram we are observed that parenchyma and pith. Okay. See, next we are coming to the secondary growth. See here. Okay. We are coming to the secondary growth. It is also very important. During the early stages of development of dipod stem, that vascular bundles are conjoint, collateral, endarc, and open vascular bundles are seen. Okay. So conjoint means what? Collateral, conjoint. The xylem and phloem together they are occurred in the vascular bundle. Endarc means what? That protoxylem and the protoxylem is inner side. Okay, so that's why endarc, and open in between the xylem and phloem, that cambium is there, that we are called that type of vascular bundles are called open type of vascular bundles. Okay, these are seen arranged in the form of a ring in the steel. Okay, see here in the first diagram you are observed that the protoxylem and primary phloem is there, epidermis, cortex, and vascular bundles. The steel is there in the first diagram A. Okay, that is the before primary anatomy of the stem. Okay, next we are coming to the at the time of secondary growth, the changes will be going on. Okay, see here. So the meristematic region present in the vascular bundle is known as the intrafascicular cambium. That inner side, intrafascicular cambium. In between the xylem and phloem, that intrafascicular cambium. The parenchyma cells of the medullary rays, which are located in between the intrafascicular cambium, become meristematic with dividing potentiality. Okay, so these newly formed strips of meristematic cells are known as interfascicular cambium. So please observe this. Whenever the cambium is present in the vascular bundle, that forms the intrafascicular cambium. So in between the vascular bundles, that medullary rays forms the interfascicular cambium. So two are combined together, that intrafascicular and intrafascicular cambium, that provides the ring. Okay, so the cells of this ring divide and gives rise two types of initials. Okay, these cells are joined with those of the intrafascicular cambium and thus organize a ring. Okay, whenever that forms the ring in the second diagram, that will be shows clearly. See here that interfascicular cambium and intrafascicular cambium together join and combine the ring. Okay, so in the third diagram, that is see here that ring, the cells of the ring divide and gives rise to two types of initials. These are fusiform initials and ray initials. Okay, the fusiform initials are elongated spindle shaped structures. Sometimes these cells may be arranged in regular rows. Such a cambium is called the stratified cambium. When the elements of the xylem and phloem are derived from such a type of cambium, those tissues also exhibit stratified condition. Example, ulmus, juglans, and robinia. Okay, so in some certain genera, the arrangement of the fusiform initials is not in a regular rows. So this type of cambium is called a non-stratified cambium. Okay, the two types of initials are there. That is fusiform initials and ray initials. But some cells are arranged in regular rows. Okay, some cells are arranged in regular rows. Such a cambium is called a stratified cambium. Some cells are arranged in not in regular rows. That type of cambium is known as the non-stratified cambium. Two types are there. So in the ray initials, all the cells are isodiametric. Secondary medullary rays are produced by division of these initials. Okay. So next, in the secondary growth, we are observed that secondary phloem and a secondary xylem are developed from that ring. Okay, so the xylem initials, the xylem going to primary xylem and secondary xylem, and primary phloem and secondary phloems are arranged. So these rays extend from the secondary xylem to the outer side of the secondary phloem. So water and food materials are transported radially through these rays. So the portion of the ray in the secondary xylem, it is called the wood ray or xylem ray. While the portion of the ray in the secondary phloem, that is known as phloem ray. Okay, this type of secondary growth we will be observed in the dicot stem, that is see here in the vascular cambium. Second is the primary phloem, and the third one is the secondary phloem. Second and third is see here, primary phloem and secondary phloem, and the fourth one is the primary xylem, and the fifth one is the secondary xylem. These are formed by the ring. Okay, so next so these rings are formed at the time of secondary growth. These are called annual rings. So the secondary xylem formed every year due to the activity of cambium, so appears like a ring in the tears of the stem. So that's why the ring which is formed in this way is called the annual ring. Okay, so yearly once rings are formed by the activity of xylem cambium. Okay, so this is the study which involves the calculation of the age of the tree by counting the number of annual rings is known as the dendrochronology. It's very important, Amma. What is the dendrochronology? Dendrochronology means what? That annual rings which involves the calculation of the age of the tree. Okay, so that that is known as the dendrochronology. It's very important. And after that, heartwood and sapwood. What is the heartwood and sapwood? In the plants, after a few years of secondary growth, the earlier formed wood will be pushed to the center. Okay, so after the secondary growth, a few years of secondary growth, the earlier formed wood will be pushed to the center by the later formed wood. By the later formed wood, that will be pushed into the center. As a result of this, part of the wood gradually becomes inactive and darker in color. So this portion of the wood is called heartwood. Okay, all are you understood? What is that? Heartwood means what? After the secondary growth, few years of secondary growth, the earlier formed wood will be the pushed to the center by the later formed wood. As a result of this, that wood gradually becomes inactive and darker in color also. So this portion of the wood is called heartwood. It is also very important. And several protrusions known as the stylosis are produced into the xylem vessels by the adjacent of parenchyma cells. Okay, so whenever the transport of the different materials takes place in the outer rings of the secondary xylem, which constitute the active part and exhibit maximum efficiency, this active part of the wood is lighter in color and is called the sapwood or alburnum. Okay, or alburnum. It's also very important. The transport of different materials it takes place in the outer rings of the secondary xylem. So this is the active part, and this wood is very lighter in color. That is called sapwood or alburnum. As the outer rings are formed in the secondary xylem gradually. Okay, that is the sapwood and hardwood. Very important. And after that, we are coming to the periderm. It's also very important. Periderm. What is the periderm? So the area of the stellar region considerably during the secondary growth due to the addition of secondary vascular tissues in the stele. Okay, secondary vascular tissues are formed when the secondary growth time, that secondary xylem, secondary phloem. Okay, as a result of this, the epidermis experiences gradual increase of pressure over it. So whenever only at the center of the stem that gradual increase of the pressure in that. So sometimes during the early part of the secondary growth, the epidermis stretches to a little extent. Okay, but later it breaks up here and there and ultimately degenerates. As a result of that, the inner tender cells of the cortex are exposed. Okay, whenever comes the pressure to the cortex, that some cells are exposed. In order to protect these cells, a secondary protective tissue is known as periderm develops in the cortex. Okay, so one of the pressure comes to from the center, so that's why that the time the cortex the cells are exposed. In order to protect these cells, that secondary protective tissue known as the periderm develops in the cortex. Periderm usually forms in the gymnosperms and woody angiosperms, but absent in the stems of monocotyledons and roots. Okay, please remember this. It's very important point. Periderm, it is usually forms in the gymnosperms and woody angiosperms, but absent in the stems of monocotyledons and roots also. Okay, periderm it's formed in the cortex at the time of secondary growth. We are observed in the only dicot stems and generally that gymnosperms and woody angiosperms, but absent in the monocot stems and roots also. Okay, three distinct regions constitute the periderm. Periderm means it having the phellogen or cork cambium, phellum or cork, and phelloderm or secondary cortex. Okay, three distinct parts we are observed in the periderm, that is phellogen or cork cambium, phellum or cork, and phelloderm or secondary cortex. You will be seeing these three distinct regions in the periderm. It's very important. What is the phellogen or cork cambium? This may develop from the epidermis or hypodermis or cortex or phloem tissue. Okay, that is the cork cambium or phellogen. The meristematic cells which constitute the cork cambium undergo repeated periclinal divisions and produce the cork tissue to the outer side. Okay, that cork tissue is to the outer side and the secondary cortex to the inner side. Okay, gradually that many periclinal divisions occur in that cells, so that will be produce the cork tissue to the outer side and the secondary cortex to the inner side. Usually more amount of cork tissue is produced than the secondary cortex. Cork cambium is a uniseriate homogeneous structure. It consists of narrow rectangular cells. Okay, so in the nerium and pyrus, the epidermal cells are differentiated into the cork cambium. It's very important. Okay, so that some important points I will give you. That in the stem of prunus, the cork cambium develops below the epidermis, which in that stem of prunus. Okay, cork cambium develops below the epidermis. In Bougainvillea and Aristolochia, the second or the third row of the cell transform into the cork cambium. Okay, Bougainvillea and Aristolochia, we are observed the second or third row of cell transform into the cork cambium. But in the stem of prunus, the cork cambium develops below the epidermis only. Okay, in the root of vitis, phellogen forms from the pericycle. This is also important thing. That is, in the root of vitis, phellogen forms from the pericycle. Okay, phellum. This is the this tissue is composed of the few layers of closely arranged cells that with intercellular spaces and with the thin cellulose cell walls during the early phase of their development. Okay, that is the phellum. So phelloderm is what? These cells have thin cellulose cell walls and are similar to the parenchymatous cells of the cortex. That is the phelloderm. Suberin is absent in their cell wall. In the phelloderm, we are observed that suberin is absent. When the phelloderm is in several rows, its cells are arranged in radial rows due to the presence of chlorophyll. These cells can perform the photosynthesis also. These are helpful in the storing of food material. Okay, that is next. What is the bark? During the secondary growth, that periderm develops in the place of the epidermis, since the cork cells are dead cells. Okay, what is that? These are dead cells. In the place of epidermis, that periderm develops the that bark. Okay, the transport of water and materials is stopped to the living cells of the outer tissues. Okay, as a result of all the cells, outer the cork become dead. The dead cells along with those of cork together constitute the bark. That is the bark. And that is what is that? Lenticels. This is also very important. So in the some due to secondary growth, the periderm develops in the place of epidermis. The calf tissue is composed of closely arranged dead suberin coated cells that is called bark. Okay, so in such an occasion that certain soil colored spots like openings, okay, so which are composed of matured cells develop over the stem. These openings are called lenticels. It is also important. These openings are called as the lenticels. These are located opposite to the stomata. Okay, lenticels. When we are observed that in the secondary growth time that some openings are there opposite to the stomata that are called lenticels. Okay, so and carry out their function in the secondary body of the plant. These lenticels do not form in certain lianas, although the periderm develops. Okay, this is the lenticels. So it is the primary structure of stem, and after the secondary growth, what parts are there? Secondary xylem, secondary phloem, periderm, and phloem, cork, lenticels. This is a important class. Sir?
Okay, madam, thank you. Thank you very much.